I would like to thank Pittcon JAIMA organizing committee for inviting me for this presentation today from Shimadzu Scientific Instruments. I would also like to thank you all for attending this presentation today. My talk entitled Analysis of Monoclonal Antibody, mAb, and Related Biologics by LC and LCMS platforms. In this talk, I'll be talking about Shimadzu's innovative technology, especially LC and LCMS platforms that can be easily applicable in your monoclonal antibody and related biologics analysis. First, I'll give a little bit overview of monoclonal antibody. Then I'll be showing LC and LCMS instrumentations for mAb analysis and quality control. For doing that, first I'll show LC methods for mAb analysis, and I'll be showing two systems. One is Nexera Bio-UHPLC based mAb analysis, and another is Nexera Bioinert UHPLC based mAb analysis. For Nexera Bioinert UHPLC is basically Shimadzu's latest innovation, which used metal-free flow path from the needle to the detector. And then I'll be showing LCMS methods for mAb analysis, and I'll be showing two technologies. One is LCMS 9030 QTOF, and another is LCMS 9060NX triple quad for mAb analysis. And finally, I'll be showing what are the take-home messages for this talk today. The typical structure of monoclonal antibody is Y-shaped, and it has Fab regions and Fc regions. In Fab regions, basically fragment regions with antigen interactions. It has variable sequence regions. On the other hand, Fc regions, which is basically fragment regions that mediates cellular effect functions, which has constant regions that defining IgG subclass. And Fab regions has three CDRs. CDRs basically complementary determining regions, and it all has connected with the disulfide bond. The red marked area here is called heavy chain, and heavy chains basically 50 kilodalton proteins contains glycan modified regions. The red marked area here is called light chain. Light chains basically 25 kilodalton protein. This is our complete portfolio of instruments that use for this study. First is Nexera Bio-UHPLC, and then Nexera XS inert or Bioinert, and then LCMS 9030 QTOF, and then LCMS 8060NX. And for the data analysis, data collections, and instrument control, we use Lab Solution CS and Lab Solution DB. That DB means database. Here I am going to show you first data which acquired by Nexera Bio-UHPLC or Nexera XSI, which is Bioinert. First, I am going to show charge variant analysis of Trastuzumab, which is one of the therapeutic mAb. Here we use four conditions. First, changing the pH, then different flow rates, different gradient time, and different column length. Here you can see when you use about 100 millimole per liter of sodium phosphate, it gives significantly separations. On the other hand, a little bit low flow rate is also gives a very nice peaks, but if we go too low, that time it didn't give the same separation. So you can see at 0.8 ml per minute, we can see the separated peak better. On the other hand, when we tried with the different gradient conditions, like a gradient time, that 5 minutes, 10 minutes, 15 minutes, and 20 minutes, we can see, and it relatively longer run give the better separations. And for column length, as all of you know, that little bit longer column gives the better separations. In this case, 100 millimeter, we got the best results. Now I am going to show protein aggregate analysis by UV-UHPLC. So we use 200 nanometers for this analysis. You can see we inject both mAbs 6 times. So they are very nicely reproducible. You can see here higher aggregates and then dimer and then monomer. For other mAbs, same thing, like higher aggregates, dimer and monomer, and they're very high reproducible. So that means the peak didn't shift from each other, which reflects that Nexera by UHPLC with Shimpact biocolumn is a useful tool to analyze the protein aggregates and fragments of this mAb. Now I am going to talk a little bit mAb analysis by Bioinert UHPLC. By Bioinert, we can do a variety of analysis like LC-SCC, then for aggregate analysis, fragment analysis, disulfide bond, methanic isomers, lysine isomers, and then glutamic acid, aspartic acid, peptide map using LCMS, and then glycan analysis using LCMS, or using LC or LCMS, we can do antibody drug or antibody drug conjugate analysis. So the reason we can do the variety of analysis without metal interactions because it has metal-free, low absorption flow path. And for the pressures, it's the outer surface is the metal that can sustain the pressure until 100 megapascal. And inside is coated with the peak that creates the metal-free flow path from the needle to the detectors. I'm going to show you some of the analysis by Bioinert UHPLC. First, mAb aggregate analysis. This is a very powerful tool for the analysis of mAb aggregates. The reason is, see, the peak is very sharp and nicely separated. So this is very useful to analysis or do the analysis for aggregates. And then if you see mAb and ADC chromatograms, upper panel and lower panel, and two types of mAb and ADC. That's one is non-conjugated mAb, that is the pure mAb, and another is the conjugated, that is antibody drug conjugates. And if you see for all of the analysis is called drug to antibody ratio analysis. Here you will see non-conjugated mAb and the lower panel is ADC. For that you can see that all the DARTs like DART 0, DART 2, DART 4, DART 6 are nicely separated and identified. That means the Bioinert UHPLC is a very powerful tool to do the drug to antibody ratio as well as the conventional mAb analysis. This is again our complete portfolio of instruments, but I am going to talk about the data analyzed by LCMS 9030 QTOF and LCMS 8060NX triple quad. We use 3 softwares for do the analysis. One is Lab Solutions DB or CS. For Lab Solution DB can be used for single instruments and single computers. But if you want to connect entire laboratory, all instruments for one server, that is Lab Solutions client server or Lab Solutions CS. So all of your laboratory instruments can be connected under the same servers. And then we used Protein Matrix softwares, that is our partner to do the protein or mAb-based analysis using LCMS. We also use one of the open source software, which is Skyline, that's for the peptide MRM analysis. This is LCMS 9030 QTOF intact NIST map. So if you zoom on of the peak, it looks like this. So that means the main peak with the glycosylated peaks. After deconvoluting that mass spectra with BIOS, you can see it's identifying with the all glycosylated peaks, and this is the LCMS chromatograms, and that's the structure of the NIST map. We also analyzed Avestin map. Here is the deconvoluted mass spectra with BIOS. You can see the peak numbers are fewer than the NIST map because this is the therapeutic map. Here is the LCMS chromatogram, and the upper is the structure of Avestin map. Now, if we put two analyses side by side, which is deconvoluted mass spectra from NIST map, on the other hand, deconvoluted mass spectra from Avestin map, if we see that you can significantly see the difference. NIST map has higher number of peaks and Avestin map has fewer number of peaks. That means by using this protocol and method, you can significantly identify and separate different type of maps from each other. We also performed map subunit analysis. To do that, we reduce the map using DTT. So DTT help reduce disulfide bond. So after the reduction using DTT, we analyze the NIST map. You can see the left panel is the light chain and the right panel is the heavy chain. They are very nicely identified and separated. We have also analyzed Avestin map. In the left panel, you can see is the light chain peaks, and the right side you can see is the heavy chain peaks. They are nicely separated and identified. After the reduction map analysis, we did the deglycosylation using PN gas. So glycan can vary indifferently on each of the heavy chains. Glycosylations play a significant role for therapeutic interventions. So that's why it is important to analyze deglycosylated peaks. Here we did same study for NIST map and Avestin map. So you can see on the upper panel that NIST map LC chromatogram and the right side Avestin LCMS chromatograms. So from there you can see the heavy chain and light chain peaks on the bottom, and those two mass spectra are quite different. So that can show which is therapeutic protein and which is just normal NIST map. To make the study complete, we are also needed to do the released glycan analysis. For after releasing the glycans, it need to be dyed with the procainamide. That procainamide helped enhance fluorescence detectability. On the other hand, it was also proven that it can significantly enhance the ionization ability in LCMS, especially using electrospray ionizations. So you can see here Avestin glycan profile on the left side and NIST map glycan profile on the right side. They both are nicely separated and you can easily quantify them. We have also did the protein digestion with trypsin for the lysine and arginine side, and here I am going to show some of the results. This is NIST map digestion with trypsin, and you can see the digestion sites, that is R and K for the light chain, and same thing for the heavy chain on the bottom panel. Similarly as before, we have tried both NIST map tryptic digest and Avestin tryptic digest. That upper panel shows the NIST map tryptic digest with all our peptide peaks, and same thing all peptide peaks for Avestin map on the bottom. After the analysis using LCMS 9030 QTOF, we load the data in protein matrix BIOS software, and it gives the NIST-mAb light chain sequence coverage, and then NIST-mAb heavy chain sequence coverage on the bottom. In both cases, the peptide mapping sequence coverage nearly 100%. We have performed similar analysis for Avastin map, and you can see the Avastin light chain on the top and Avastin heavy chain on the bottom. In both cases, the peptide mapping sequence coverage were nearly 100%. I am going to show you selective digestion of IgGs by nSMOL. nSMOL is basically Shimadzu's proprietary technology, that is nano surface molecular oriented limited proteolysis. If you have any protein that has significantly low abundance, that can be enhanced using this protocol. So how we can use this? So first, let's say it's a mAb. So mAb has Fab and Fc regions, as I have shown earlier. So Fab region is oriented towards the solutions. So Fc is captured with the beads, and then it's released with the digestion and then analysis. In the next slides, I am going to show you how the analysis can be done. So here you can see that first, IgG fractions by protein resin from diluted plasma you incubate for 15 minutes and then put for resin wash for 5 minutes and then load the Fg trypsin nanoparticles on that same solutions. After 5 hours of digestion, do the 5 to 7 minute runs, and then you can analyze for the MRM based quantities or which way you want. So now I am going to show bevacizumab's minimal Fc peptide by nSMOL. So as mentioned earlier, the Fc regions can be significantly enhanced. So here is the highest one, but the bottom one was really low in the normal analysis. But when we did the analysis using nSMOL, it significantly enhanced for the identifications and quantitations. Here I am going to show MRM based quantitation of bevacizumab using nSMOL. The lowest peak was, that means the lowest concentrated peak was 10 nanogram per ml. You can see the peak is nice shaped, and signal to noise ratio is 20, and this is the blank. And after the analysis from 10 nanogram per ml to 10,000 nanogram per ml, you can see that calibration curve was obtained with the R-square value of 0.99, and the signal to noise ratio of the lowest peak was greater than 20. So this is a very good quantitations and the calibration curve obtained using this protocol. Now I would like to summarize my talk. What will happen if we put everything together? Let's think you have a bioprocess analytics and a QC lab. You want to do only LC based analysis. You can use Nexera, XSI, Bioinert, UHPLC for oligonucleotide, monoclonal antibody, or any kind of biologics analysis without any compromise. Now you are thinking you want to do peptide MRM based quantitation analysis for your digested peptide or any kind of peptides. In that case, LCMS 8060NX would be highly beneficial. Now you are thinking you want to do peptide mapping or intact protein analysis or even N-glycan analysis. In that case, Shimadzu's LCMS 9030 QTOF high resolution mass spectrometry would be very useful. And all those analysis we use BIOS protein matrix softwares from our partner. If you do all those things, you will have consistent, high quality biopharmaceutical analysis. And now you are thinking your Fc regions of your map is significantly low. You can enhance your Fc regions by using Shimadzu's proprietary analytical technology or kit, that is nSMOL. Now you have a lab, you can run entire lab with our lab solution CS database or client server. And in that way, you can really make a nice laboratory for protein, mAb, or any kind of biologics analysis. Now I would like to thank and acknowledge who helped to complete these studies. From Shimadzu Scientific Instruments, Stephen, Vicky, Chris, Evelyn, Takashi, and Takashi, and from Protein Matrix, St. John's and Michelle, and then from Intertech Pharmaceutical Services, Michael, Dale, Lawrence, Reed. Thank you all for your great help. Finally, I would like to thank you very much for your kind attention. For any additional information or questions, please send me an email. My email address is mkmandal@shimadzu.com. Please visit our website www.solutionsforpharma.com. You will be amazed by knowing our technology, that how Shimadzu can help enhance your laboratory's biologic analysis by providing artificial intelligence, data analytics, latent instrumentations, and automations. And again, thank you very much for your kind attention.